if I draw the cell membrane like this, its cell membrane somewhere in between will develop in growths like this. So similar type of growths are present. This resembles a lot with the bacterial mesosome. This structure in the bacterium is called as the mesosome. Functions and all we'll be learning about in later classes. So the first difference between the outer membrane and inner membrane is outer membrane is smooth, inner membrane is infolded. It is folded towards the inside or invagination, and you call these infoldings as crista. Now the permeability of the outer membrane is more compared to the permeability of the inner membrane. It is less permeable and more selective in nature. There are porins present here. What are porins? Remember, transport proteins are of two types. Tunnel or the tunnel protein, or otherwise you can even call them as the porins because they allow things to go across, or otherwise channel protein. And the other type of protein which I told you in that classes were the carrier proteins. Carrier proteins take a round of 180 degree and release the content on the opposite side. So what I am trying to tell you here is that the outer membrane carries the proteins which are just like a tunnel, but the inner membrane carries the carrier proteins. Oxysomes are absent. Oxysomes we'll be learning within few minutes later. So oxysomes are absent in the outer membrane, but they are present in rich number in the case of the inner membrane. Now the composition. Outer membrane is 60% 60% proteins and 40% lipids in composition, and the lipids basically are phospholipids and cholesterol. But when you see the inner membrane, it carries more amount of protein and quite less amount of the lipids in it, and the amount of phospholipid and cholesterol, that means, is less. ETS, which you will be learning in the respiration in the plant physiology quite later in the 11th class. ETS is absent. Full form is electron transport system or electron transport chain, ETC, means the same. So ETS or ETC is absent in the case of the outer membrane, but it is present in the case of the inner membrane. So there are huge number of differences in the outer and inner membrane. Why I discussed this? Because within few minutes you're going to understand something very much new. Now the next thing is what scientists basically believe is that the mitochondria and chloroplast also, I'll be talking about that's next topic only. So mitochondria and chloroplast, they are something like parasites. Now the whole history goes like this, that the science today believe that this was explained by Altman and Schimper in 1890 and 1896. They stated that the mitochondria, including the chloroplast, so mitochondria was a parasite which was an endoparasite, so it used to enter into eukaryotic cells and then cause problem to them. Slowly this parasite became a permanent member of inside the cell of a eukaryotic organism. Now this theory has been confirmed because of the many evidences. Now the first evidence. The outer membrane, if you see, is cellular in origin. Its composition is quite similar to the plasma membrane. But when you look at the inner membrane has enzymes. I told you there are enzymes which are called as oxysomes, and there are enzymes which are called as ETS. So these two things are enzymes. And if you pay attention to the bacterial cell structure also, its membrane carries enzymes. So membranes carrying enzyme is a prokaryotic feature. So this made scientists think in this manner, that there was a bacteria which has entered inside the bacteria, means a mitochondrial bacteria or prokaryotic structure, which entered inside and became a endosymbiont. Symbiont means mutualism. That means both of them are helping each other and helping in survival. So it has become an endosymbiotic in origin. And one more thing, mesosomes are there. Another important thing, the ribosomes which are present inside the mitochondria are of which type? Prokaryotic type. You can see the numbers. So the mitochondria present inside the chloroplast, even same. We are talking mitochondria right now. So the mitochondrial ribosomes are similar in its sedimentation coefficient, or Swedberg's unit is exactly similar to the prokaryotes. This also gives us an idea that maybe these structures are prokaryotic in nature and they have become endosymbionts inside the eukaryotic cells. Now another point to confirm this, mitochondria carries its own DNA. So there is no other organelle apart from chloroplast again. So mitochondria carries its own DNA. The DNA can be linear, basically in the case of the plant's mitochondria. It would be circular in the case of the animal mitochondria. And the mitochondrial DNA carries more amount of GNC. Now what is GNC? Again, something in the biomolecules, or next chapter we are going to learn. DNA and RNA carries